Imagine your immune system as an army. Every soldier needs two things: a place to be born and trained, and a place to actually fight the enemy. Today, we are touring both the training academies and the battlefields. Yes, you heard it right. The training academies are nothing but we will be learning about the primary lymphoid organs, and the battlefield is nothing but we will be learning about the secondary lymphoid organs. Right. So now we will be learning about the two camps here, the primary and the secondary lymphoid organs, which play a major role in our immune system. So are you all ready with me for BioBytes next class? Welcome. So let us start. Now, the primary lymphoid organ. The first primary lymphoid organ we will be learning about is thymus. Right? The second one is the bone marrow. First, let us learn about thymus. Now, the thymus, it is a place, as I have told you, so it is a place where the soldiers are born and trained. And you can see thymus, if you see, it is a pyramid shaped asymmetrical bilobed gland. You can see it is having two lobes, right, and it is located between the breastbone and the heart. It is located between breastbone and heart. It has two lobes, and these two lobes are connected by a structure called as isthmus. So the isthmus connects both the lobes. And the thymus, it is very active in childhood, but atrophies with age and is replaced by fat. Right? We also call it as thymus involution. Involution. So thymus involution means this thymus gland is very active during childhood, right? But as a child grows older and older, this thymus gland is completely replaced by fat, right? So now we call it as thymus involution. Now if you see, a thymus is divided into many lobules, right? And this lobule is covered by capsule. You can see this is one lobule what we are delivering. So this is a bilobed thymus gland, right? And this is a lobule. Now this lobule, you can see it is surrounded by the capsule, right? So the capsule, it surrounds the lobule, and you can see the capsule, it sends many small structures here. These structures what you see are called as trabeculae. Trabecula is singular, trabeculae. Now this trabeculae divides the tissue into incomplete lobules inside. Now each lobule, if you see here, it consists of two zones. So each lobule, so this is a histological view of one lobule, right? So each lobule shows two zones. The outer zone is called as the cortex, and the inner zone is called as the medulla, right? The light inner zone medulla and the dark outer zone as cortex. This is the structure of thymus, right? Now if you see the function, the thymus, it produces a hormone called as thymosin. There are other hormones also, but the important one is thymosin, which helps in the development of T cells. So T cells mature here. Actually the T cells are formed in the bone marrow. From the bone marrow you can see the immature thymocytes, or we call them as prothymocytes. The immature thymocytes, they are sent to the thymus. There in the thymus the T cells mature, and these mature T cells are sent into the bloodstream. So T cell maturation mostly occurs in the thymus, right? So this is about thymus. It is a primary lymphoid organ. It is located in the chest cavity. It is bilobed, right? It has cortex, which the cortex or the outer dark part consists of T cell precursors. Precursors means the ancestors like, and these precursors enter into the medulla. In the medulla they mature into T cells, and the Hassel's corpuscles, right, which are the most important parts of the immune system, and they regulate the cell mediated immunity. So this is about thymus. Now let us move on to the next one. We call it as the bone marrow. Now the bone marrow is a primary lymphoid organ. You can see the bone marrow, it is a primary lymphoid organ which is present in the medullary cavity of bones. So this is the bone marrow, right? So you can see the bone marrow. Now there are two regions of the bone marrow, as you can see. You have a vascular region which consists of lot of blood vessels, right? You can see the vascular region and the non-vascular region, right? The non-vascular region contains the stem cells and supportive cells, and this non-vascular region is a site of hematopoiesis. So hemato means blood, poiesis means generate or formation of blood cells occurs here in the non-vascular section. Now in the vascular section you have all the mature blood cells which are formed in the non-vascular section. They are sent into the vascular section, right? Now if you see in the stem cells or in the bone marrow, we have two types of cells, the hematopoietic cells and the mesenchymal cells. These are the two types of cells present in the bone marrow. Now the hematopoietic, hem means blood. The hematopoietic stem cells, right, they give rise to all types of blood cells, that is the RBCs, the WBCs and the platelets. All type of blood cells form from the hematopoietic stem cells, whereas the mesenchymal stem cells give rise to the connective tissue, that is the fat cells, the cartilage cells, the bone cells and the fibrous connective tissue. So they give rise to non-blood components, and the hematopoietic cells give rise to all the types of blood components. So these are the types of cells we see in the bone marrow. So the hematopoietic stem cells and the mesenchymal stem cells. Now hematopoietic stem cells, they produce all type of blood cells and they maintain the constant supply of blood cells, as we have learned. Now the non-hematopoietic stem cells or the mesenchymal stem cells produce the non-blood components. So if you see the hematopoietic stem cells, again they divide themselves into myeloid progenitor and lymphoid progenitor. Now the myeloid progenitor means a precursor. Precursor means the ancestor, the one which gives rise to that particular blood cell. So the myeloid progenitor gives rise to RBC, platelets, neutrophils, eosinophils, basophils and monocytes, whereas only one particular cell is here missing, that is a lymphocyte, right? So which the hematopoietic stem cells, they again are divided into lymphoid progenitor cells, which give rise to the lymphoid cells, lymphocytes, that is the B cells, T cells and NK cells, the natural killer cells, right? So hematopoietic stem cells, they are again divided into myeloid progenitor and lymphoid progenitor. Myeloid will give rise to all type of blood cells except the lymphocytes, which are produced by the lymphoid progenitor, right? The lymphoid progenitor will form the B cells, the T cells and the NK cells, right? So you should know the fact that the red bone marrow is active in children and in flat bones, whereas flat bones, that is a end of long bone, the skull and the end of long bones, right? So now let us move on to the secondary lymphoid organs. So primary lymphoid organ was the thymus and bone marrow. The secondary lymphoid organ is where they really act. So the secondary lymphoid organs are the organs where the battle happens. Now, so here once the uh, T cells and B cells are trained, they don't stay in the academy of primary lymphoid cells, right? Primary lymphoid organs, they don't stay there, they patrol the body waiting at the checkpoints. Now what are the checkpoints? The checkpoints are the secondary lymphoid organs, also known as the peripheral lymphoid organs. These organs are mainly responsible for antigen specific immune responses, right? Where this is the place where antigen antibody reaction takes place. Now, the secondary lymphoid organs are mostly the lymph nodes and the spleen. Now, these lymph nodes, again you see children, these secondary lymphoid organs are divided into encapsulated, encapsulated and non-encapsulated. Encapsulated means which are covered by a capsule or which have a lining, whereas non-encapsulated means they don't have a particular lining, they are directly in contact with the antigens. So the lymph nodes and the spleen comes under encapsulated lymphoid organs. Now if you see the lymph nodes, they are small bean shaped structures, they are present throughout the body, they are mainly found in the neck, underarms, abdomen and groin. So you know very well, groin is the area where our the leg portion starts. That means between the lower abdomen and the starting of the leg or the thigh bones. That area we call it as groin. So here we have the lymph nodes, and these lymph nodes are richly supplied by lymphatic vessels and blood vessels, and they help in the entry and exit of lymphocytes. Now if you see how a lymph node looks like, lymph node has three areas. It has cortex, it has paracortex and it has medulla, right? So cortex, paracortex and medulla. In this, paracortex is the area where there is lot of T cell activity going on. In the cortex, the lymphoid follicles are present. You can see the lymphoid follicles here, which produce the lymphocytes are produced here, and lymphocytes, they mainly act upon here in the cortex region. And you have the rich supply of blood vessels which bring all these cells for the interaction. Now next we have, now the functions of lymph nodes is, it is a site of antigen dependent proliferation. It is a differentiation of specific lymphocytes, the lymphocytes like the T cells, the B cells and the natural killer cells, and here filtering and trapping of antigens takes place here. So this is, as I told you, this is a place where the battlefield, so where the antigen and antibodies react. Then next we have spleen. So spleen is a fist sized organ located in the left upper abdomen under the ribs. So it is also known as the immunologic filter of blood. So lymph is also known as immunologic filter, and remember, spleen is also known as the graveyard of RBC, because our RBCs, they live only for 120 days, their lifespan is 120 days, and after 120 days all the RBCs, right, they are brought here and they are they are put up here, all the dead RBCs. That is why we also call it as graveyard of RBC. So if you see this spleen, it is divided into again two zones, the white pulp zone and the red pulp zone. The red pulp zone has lot of blood vessels, and the white pulp zone has some lymphoid sheaths. You can see the lymphoid sheaths, sinus, sinusoids, periarteriolar lymphoid sheaths. So the marginal zones, these are all form a part of white pulp, that is a non-vascular region, and the red pulp forms the vascular region. So what is the importance of this? The red pulp, it is rich in blood cells, the macrophages, lymphocytes and other cells, whereas the white pulp contains lot of lymphocytes which mainly play a major role in immune responses against antigens. Here where the formation and maturation of blood cells takes place, here you have the real immune responses against antigens. Now if you see the function of spleen, spleen plays a major role in filtering the foreign material, right, from the blood. What all foreign bodies are coming, those are filtered by the spleen. The macrophages and dendritic cells present present antigen to B or T cells. As I told you, the T cells will present the antigens to the B cells, and the B cells finally produce the plasma cells which fight with the antigens. So all that happens here. The B cells get activated and produce large amounts of antibodies here, and the old RBCs are also destroyed in the red pulp, as I have already told you. Now, in addition to this, right, we have learnt about the lymph nodes and the spleen, which are called as encapsulated lymphoid organs. The non-encapsulated lymphoid organs are known as the MALT. We call them as MALT. MALT stands for MALT. MALT stands for mucosal associated lymphoid tissue, right? So MALT stands for mucosal, that means where all you have the mucus, right? Mucosal associated lymphoid tissue. So this MALT, they are present in different parts of the body, right? Like if they are present in the gut, mostly in the small intestine, in the small intestine, in the ileum part, we call them as Peyer's patches, right? So galt is a gut associated lymphoid tissue. Here they will fight, here they act as, the role of this malt is they act as immunity, like they play a major role in immunity, so fighting against the pathogens here. So in the gut you have the galt, which is a gut associated lymphoid tissue. In the respiratory tract you have BALT, bronchial associated lymphoid tissue. In the nasal area you have the NALT, nasal associated lymphoid tissue. In the vulvovaginal area you have the VALT, vulvovaginal associated lymphoid tissue, right? You have the larynx associated lymphoid tissue, you have the skin associated lymphoid tissue, and you have the conjunctiva associated lymphoid tissue. So you can see all these are the areas where the infection, the chance of infection is more, right? So you have all these non-encapsulated lymphoid organs. You have here the tonsils. Now tonsils is a big process going on, which is still under research. Some scientists consider tonsils as encapsulated, some consider it as non-encapsulated. It is still an area of debate. But these tonsils, they are present in the at the back of the tongue, at the sides, pharyngeal or nasopharyngeal, and near the buccal area. So these tonsils also come under the non-encapsulated lymphoid organs, as most of the scientists believe. Right. Now, so it's done with our uh, primary and secondary lymphoid organs. Let us practice what we have learned. So we have the challenging MCQs here to test our understanding. Which of the following best explains why thymic involution? I told you what thymic involution is. Thymic involution means it is very active during childhood, but as the child grows older and older, it has a deposition of fat. So thymic involution does not completely eliminate T cell mediated immunity. As we have learned, thymus plays a major role in activating the T cells, maturing T cells. But when thymic involution occurs, how come the T cell mediated immunity is seen in adults? So the reason is the mature T cells generated earlier in the life persist as long lived memory or peripheral cells, because during childhood, when the thymus mature, the T cells which get matured, they are actually long lived, and they are very much capable of dividing and giving rise to more T cells. So they are actually long lived. But you see, after certain time in old age, this long lived T cells decrease, and that is where the immunity of the person as he grows older and older also decreases. So it is all the question of thymic involution. So this is not the answer, because thymus regenerates fully during infection, no. Bone marrow takes over T cells, no. T cell maturation occurs only in the thymus, not in the bone marrow. Lymph nodes produce new T cells, no. So the correct answer is this one. The Hassel's corpuscles found in the thymic medulla are primarily associated with, so Hassel's corpuscles. Now Hassel's corpuscles, children, you need to remember that they are not responsible for antibody production. Antibody production, it is a plasma cells. RBC destruction, no. Antigen trapping, no. It is again plasma cells. Negative selection of self-reactive T cells. Now what do you mean by this self-reactive T cells? Self-reactive T cells are nothing but there are some T cells which before they are put into the blood cells, and before they start working, they start identifying any self body, any self protein, and they start acting against that protein which is its own self. So such self reactive T cells are identified and they are killed here. So that is what we call it as negative selection of self reactive T cells. So such T cells, before they enter into the bloodstream, they are identified and they are eliminated. So that is the Hassel's corpuscles are mainly associated with the negative selection or the self reactive T cells. Then next, which region of lymph node contains T cells and dendritic cells, and functionally analogous to which region of the spleen? So the T cells, we have the paracortex or the white pulp. So they are mostly related to the T cells, because the cortex region, the cortex and the medulla region, they are related to the B cells, the cortex region, whereas the T cells are related to the paracortex region. Right. So now here the next, the splenectomized, that means splenectomized patient is whose spleen is removed, is significantly higher risk of infection, primarily from which category of pathogen? So this, if a patient's spleen is removed, he has a higher risk of infection, primarily from bacteria, right? So bacteria, it's not the viruses, fungus or protozoan, it is mainly from the bacteria. Then next, why are primary lymphoid organs considered essential for central tolerance while secondary lymphoid organs are not? Right. Now, central tolerance means elimination of self reactive lymphocytes. So, primary lymphoid organs are considered essential for central tolerance. So, in the secondary lymphoid organs, you have only the war going on. But in primary lymphoid organs, you have the main, they are born here. So, the elimination of self reactive lymphocytes occurs during lymphocyte maturation, which happens exclusively in the primary lymphoid organs, right? So secondary, this cannot be the answer. Secondary organs lack any immune cells, no. In secondary lymphoid only we have the immune cells fighting, and central tolerance applies to B cells which mature in secondary organs, so they don't mature here. Here you see central tolerance only applies to B cells, no, it doesn't apply to only B cells, even for the T cells. Here only primary organs contain antigen presenting cells, no. Antigen presenting cells, they are mainly produced in the secondary lymphoid organs, there they fight. So the answer is B, central tolerance occurs during lymphocyte maturation, which happens exclusively in primary organs. Now, which of the following is classified as a non-encapsulated secondary lymphoid tissue? Very easy, right? So, non-encapsulated spleen, lymph node, right? These are tonsils. It is also considered to be, it is still a question mark. So, very clear answer is given here. Gut associated lymphoid tissue, it is non encapsulated. All the associated lymphoid tissue, MALT, mucosal associated lymphoid tissues, they are non encapsulated. Right. Bone marrow mesenchymal stem cells differ from hematopoietic stem cells in that they primarily give rise to. So, mesenchymal stem cells, we have hematopoietic stem cells which give rise to blood cells and platelets. Hematopoietic stem cells, mesenchymal stem cells, we have learned they give rise to all the other connective tissue, the fat, cartilage, bone, and fibrous connective tissue. Right. Now, in the germinal layer of a lymph node follicle, which process is primarily occurring after antigen stimulation? Germinal center of lymph node follicle, which process is primarily occurring? So, T cell negative selection. Children, T cell negative selection occurs in the thymus, right? Hematopoiesis occurs in the bone marrow. RBC destruction occurs in the spleen, right? So the germinal center of a lymph node follicle, right, it occurs in the B cell proliferation. So the answer is B. So this was all in our primary and secondary lymphoid organs. Keep learning. Thank you.